Hi, my name is David the Hahn, and I've traveled the globe searching for cells. Crikey! Today we observe a natural cell in its wild habitat. Look at its beauty. It's just grazing over there. We're gonna watch it go through the process called meiosis, in which it turns from a diploid cell to 4 haploid cells. We are now going to creep up on this grazing sac cell. Stop. It senses fear. It feels it's necessary to duplicate itself in order to protect itself from any danger. Before dividing, the cell must go through the necessary steps, the first of which is called prophase 1. It now must pack up all its loosely wound chromatin into chromosomes. Crikey! The nuclear envelope will now break up. The nucleolus will now proceed to disappear. Alright. Crikey! Where did it go? Now, the 2 homologous chromosomes will pair up to form a tetrad, being held together by a protein structure called a centromere. Well, just look at that centromere. First, the strands of DNA break,
They're making me wear a snake.
but then eventually reattach.
I told them I didn't want.
Now, the sister chromatids intermingle and share genetic material at a place called the chiasma. Now, the spindle fibers attach to each homolog. Now, the homologs will line up in the middle of the cell. Now, during anaphase 1, the homologs will migrate to opposite ends of the cell. Now, during telophase 1, the spindle fibers will now disassemble. The nuclear envelope and the nucleolus will now reappear. Alright. During cytokinesis, the cell will now split into 2, leaving 2 haploid cells. This process repeats itself, and we will now end up with 4 haploid cells. Now, wasn't that fascinating? This has been Cell Hunter, brought to you by David the Hahn and Crocodile Dundee.
The Golgi apparatus. It's the Golgi apparatus. Yes, the Golgi apparatus. Don't care who's staring at us in the Golgi apparatus. It modifies proteins and moves lipids around and packages things to keep them safe and sound.